Hello everyone, my name's Iman. Welcome back to my YouTube channel. Today we're going to cover chapter 4 in our MCAT Biochemistry series, and this chapter is titled Carbohydrate Structure and Function. In this chapter, we're going to cover the following objectives. First, we're going to start with carbohydrate classification. This means we're going to go over how carbohydrates are named and how we use stereochemistry to distinguish between different isomers. Second, we're going to talk about cyclic sugar molecules. Here, we're going to explore how ring formation happens and what conformations are stable. We're also going to touch on the concept of mutarotation, which is where one form of a sugar can convert into another in solution. Third, we're going to look at monosaccharide chemistry. That includes oxidation and reduction reactions, ester formation, and how sugars form glycosidic bonds when they react with alcohols, which is really important for building larger carbohydrates. Finally, we're gonna wrap up this chapter with a discussion on complex carbohydrates. We're gonna study disaccharides like sucrose, lactose, and maltose, and then we're gonna move into polysaccharides like starch, cellulose, and glycogen. With this introduction in place, let's move into objective 1 and talk about how we classify carbohydrates. Carbohydrates come in many forms, but they all follow a few structural patterns that allows us to categorize them. When we talk about classification, we're usually referring to 4 main features. The first feature is the number of sugar units in the molecule. The second feature is the number of carbon atoms in each sugar unit. The third feature is the type of functional group that's present, either an aldehyde or a ketone. And the fourth feature is the molecule stereochemistry, or how its atoms are arranged in space. Let's start with the number of sugar units. If a carbohydrate contains just one sugar unit, it's called a monosaccharide. These are the simplest sugars, and they are the building blocks for larger carbohydrates. If we have 2 linked sugar units, they form a disaccharide. And when we have many sugar units that are linked together, we call it a polysaccharide. So depending on how many sugar units are present, we classify carbohydrates as mono, di, or polysaccharides. Next, we can classify them by how many carbon atoms are in the backbone of each sugar. A sugar with 3 carbon atoms is called a triose. 4 carbon atoms is a tetrose. 5 carbons, pentose. And 6 carbons, that's a hexose. And hexose, this is one of the most common types in the body. So for example, glucose is a hexose. Now let's talk about functional groups. These are very central to sugar chemistry. All monosaccharides contain a carbonyl group. A carbonyl group is a carbon atom that is double bonded to an oxygen. The type and the position of this group determines whether the sugar is an aldose or a ketose. If a sugar contains an aldehyde group as its most oxidized functional group, it's going to be classified as an aldose. If it contains a ketone group as its most oxidized functional group, then it's called a ketose. In addition, aldehydes occur when the carbonyl group is on the end of the carbon chain. That carbon is going to be double bonded to oxygen, and it's also going to be bonded to a hydrogen atom. Ketones, on the other hand, occur when the carbonyl group is located on an internal carbon. So that carbon is also going to be bonded to 2 other carbon atoms. Let's now look at 2 examples. Here on the left, we have glyceraldehyde. This is the simplest aldose. You'll notice it has 3 carbon atoms, which makes it a triose, and its carbonyl group at the top of the chain is an aldehyde. So this is a aldotriose. Then on the right, we have dihydroxyacetone. This is the simplest ketose. Notice it also has 3 carbons, but the carbonyl group is in the middle carbon. So this is a keto-triose. Beyond this, there are also a handful of sugars that you're expected to recognize by their common names. These include D-fructose, D-glucose, D-galactose, and D-mannose. Now, you don't need to memorize every detail about every sugar, but you do need to be able to recognize these structures and know how they relate to one another. So let's go through each one. Starting on the left, we have D-fructose. Notice that it contains a ketone group at the second carbon instead of an aldehyde at the top. That makes fructose a ketose. Specifically, it has 6 carbons, so we can better classify it as a ketohexose. Next is D-glucose. This is one of the most important sugars in the human body. You should commit this structure to memory. Now here, we have 6 carbons, and there is an aldehyde functional group as the most oxidized group present, so D-glucose is an aldohexose. The location of the hydroxyl groups on carbons 2, 3, 4, and 5 are really important to remember. It follows the pattern right, left, right, right. This is going to help you compare it to other sugars like D-galactose. D-galactose is very similar to glucose, but it differs only at carbon 4. If we look at carbon 4 in D-glucose, the hydroxyl group is on the right. If we look at carbon 4 in D-galactose, the hydroxyl group is on the left. And that is the only difference between the two, and that makes them epimers. More on that soon, but keep that word in mind. Finally, we have D-mannose. Again, this sugar is really similar to glucose. The only difference is at carbon 2. If we look at carbon 2 in D-glucose, the hydroxyl group is on the right. If we look at D-mannose, the hydroxyl group is on the left. And that is the only difference between these two. That makes them epimers as well. Again, keep that word in mind. Now that we've seen some of these common sugars that we should be familiar with, let's go ahead and talk about how these molecules can differ in their spatial arrangement. So in other words, let's discuss stereochemistry. Carbohydrates often exist as stereoisomers, also known as optical isomers. These are molecules that are going to have the same molecular formula and the same atoms connected in the same order, but they're only going to differ in how those atoms are arranged in 3-dimensional space. And that difference can really have major effects on how the molecule behaves in biological systems. To really understand stereochemistry, the first thing that we need to be able to recognize is a chiral center. A chiral center is going to be a carbon atom that is bonded to 4 different groups. When you see a carbon, like that in the sugars we saw earlier, it tells you that that molecule can exist in multiple isomeric forms depending on how those groups are arranged around that center, right? These sugars that we've been looking at, they have chiral carbons. And again, that tells us how many different isomeric forms of this sugar can exist. Now, your first step when you're analyzing a sugar should always be to count the number of chiral centers. That's going to help you figure out how many stereoisomers are possible. The general rule is that the number of possible stereoisomers is 2 to the power of n, where n is the number of chiral centers. Now, from here, we can distinguish between different types of stereoisomers. We can discuss enantiomers and diastereomers. Enantiomers are pairs of molecules that are non-superimposable mirror images of each other. That means they are mirror images, but no matter how you rotate them, you can't align them to be identical. And a good example of this concept is looking at your hands. Your left hand and your right hand, they're mirror images of each other, but when you overlay them, you'll notice that they're non-superimposable. So this is what we mean when we define enantiomers. In addition to be enantiomers, molecules have to differ at every chiral center. Now, in contrast, we have diastereomers. These are stereoisomers, but they're not mirror images. They're going to differ at one or more chiral centers, but not all. So they're non-identical and not mirror related. Within this category, there's a special subtype called epimers. These are diastereomers that differ in configuration at only one chiral center. Emphasis on the one chiral center. A good example is glucose and galactose, or glucose and mannose. Now, some of you may remember that in our MCAT organic chemistry playlist, we learned the R and S system for absolute configuration. That system assigns priority to groups around a chiral center based on atomic number and spatial orientation. And we also learned how to use that system and that information to distinguish between enantiomers and diastereomers. If you haven't watched that, I recommend. It's chapter 2 in our updated MCAT organic chemistry playlist. Now here in this biochemistry chapter, we're actually going to cover and use a different system, the D and L naming convention. So that's what we're going to focus on here. Now, the DL system, it's based on the molecule's structural similarity to D-glyceraldehyde and L-glyceraldehyde, which are simple 3-carbon sugars. So the DL system, it's all about relative configuration. And here's how it works. You're going to look at the chiral carbon that's the farthest from the carbonyl group. If the hydroxyl group on that carbon points to the right in a Fisher projection, the molecule is classified as D. If it points to the left, it's going to be classified as L. So right means D, left means L, and that's it. It's as simple as that. Now this system, again, it applies specifically to Fisher projections. These are standardized 2D representations of molecules that are used to convey stereochemistry. And as a reminder, in a Fisher projection, the vertical lines represent bonds that are going into the page. So these are drawn like dashed lines. And the horizontal lines, those represent bonds coming out of the page. So we draw them as wedges. So whenever you look at a Fisher projection, remember, vertical is into the page, horizontal is out of the page. Now let's take D-glucose as an example. If we look at the chiral carbon, that's the farthest from the carbonyl, which is carbon 5 in this case, the hydroxyl group is on the right. That makes it a D sugar. If we were to look at the mirror image of D-glucose and look at carbon 5, you will notice that the hydroxyl group is on the left, and that makes it L sugar. So these two molecules are mirror images and they're non-superimposable, and we looked at the farthest carbon from the carbonyl group to assign them D or L assignments. Now, it's worth noting that most naturally occurring sugars in the human body are D sugars, and most naturally occurring amino acids are L amino acids. So just to summarize once more, you find the chiral carbon, that's the farthest from the carbonyl, and you're going to check whether the hydroxyl group, the OH group, is on the right, D, or on the left, L, in the Fisher projection. And that's it. That's your classification. We are now all done with Objective 1, and let's move into Objective 2 and talk about what happens when sugars form cyclic molecules. Because in aqueous solution, that's actually how most monosaccharides exist. Monosaccharides can undergo intramolecular reactions to form cyclic hemiacetyls and hemiketals. And when we say intramolecular reaction, we mean a reaction that's happening within the same molecule. This occurs when a hydroxyl group on one carbon acts as a nucleophile and attacks the carbonyl group elsewhere in the same molecule. The result is a folded closed ring structure. But not all ring sizes are equally stable. Because of ring strain, only certain rings tend to form under physiological conditions. The most stable are 5-membered rings and 6-membered rings. A 5-membered ring is called a furanose, and a 6-membered ring is called a pyranose. Now let's keep this in mind and take a look at D-glucose as an example. In its linear Fischer projection, glucose contains an aldehyde at carbon 1, and then it has hydroxyl groups on several other carbons. When it cyclizes, the hydroxyl group at carbon 5, it attacks the carbonyl carbon at carbon 1, and it closes the ring and forms a 6-membered pyranose structure. Now let's number this linear Fischer projection. This carbon is carbon number 1. This is 2, 3, 4, 5, and 6. This hydroxyl group at carbon 5 is going to attack carbon number 1, and this double bond is going to break, and we're going to get this pyranose structure. In this pyranose structure, this is carbon number 1, 2, 3, 4, 5, and 6. Now, the bond that forms between the oxygen on carbon 5 and the former carbonyl carbon creates a hemiacetal. And with this numbered, let's relate it to the Haworth projection. In this projection, this is carbon number 1, 2, 3, 4, 5, and 6. Now, the carbon that was originally part of the carbonyl group, that's going to be carbon 1 in the Haworth projection, this creates a new chiral center, and we actually give it a special name. It's called the anomeric carbon. With that being said, we have to consider the position of the newly formed hydroxyl group at that anomeric carbon. It actually gives rise to two different cyclic forms of D-glucose, the alpha anomer and the beta anomer. In alpha D-glucose, the hydroxyl group on the anomeric carbon points down in the Haworth projection. That places it on the opposite side of the ring as the CH2OH group on carbon 5. In beta D-glucose, that same hydroxyl group points up, and it's on the same side of the ring as the primary alcohol group on carbon 5. Now, to tie together some terms here, you've noticed that anomers, they differ only in their configuration at the anomeric carbon. Because of that, it makes alpha D-glucose and beta D-glucose epimers as well. From here, I want to take a brief moment to explain to you how we can convert from this Fischer projection to this Haworth projection. The first thing you want to do is you want to place your oxygen right here and then draw a 6-membered ring. We have our oxygen in this corner, then carbon 1, 2, 3, 4, and 5. At carbon 5, the CH2OH group, it is always placed pointing up. Then, any group on the right side of the Fischer projection, that's always going to point downwards. And any group that's placed on the left side of the Fischer projection, that's going to point up in the ring. So let's work through carbons 2 and 3 and 4. That's what we're referring to when we say these rules for this conversion. At carbon 2, our hydroxyl group is on the right, our hydrogen group is on the left. Whatever's on the right points down, whatever's on the left points up. Then we do the same thing at carbon 3. We have a hydrogen on the right, a hydroxyl group on the left. So the hydrogen is placed down, the hydroxyl group is placed up. And then the same applies at carbon 4. And that's how we convert from this Fischer projection to this Haworth projection. Now that we've seen how to draw these structures, let's return to the distinction between alpha and beta anomers. Again, the difference comes down to the position of the hydroxyl group on the anomeric carbon after the ring is formed. And I'm reiterating this because it's super important. So in alpha D-glucose, the hydroxyl group is pointing downward. It's going to be opposite to the CH2OH group. I've also drawn the same structure in its chair conformation so that you can explicitly see that difference. In beta-D-glucose, that hydroxyl group on the anomeric carbon, it points upward. It's on the same side as the CH2OH group. And once again, I've drawn the chair conformation of that structure so that you can see what I mean. Now, here's something that's really interesting. Once the ring is formed, these two forms can actually interconvert. This is a process called mutarotation. Mutarotation occurs when the rings open up into the linear form and then reclose. When it does, the OH on the anomeric carbon, it can flip and it can switch the sugar from the alpha to the beta or beta to the alpha form. This interconversion happens spontaneously in water, and it's even faster in the presence of an acid or a base. In aqueous solution, beta-D-glucose is favored because the hydroxyl group at the anomeric carbon, it occupies an equatorial position in the chair conformation, and that leads to less steric hindrance. However, in the solid state, alpha-D-glucose can sometimes be more stable, and that's due to the anomeric effect. This is an electronic interaction that favors axial positioning of electron-donating groups next to the ring oxygen. We've now completed Objective 2, and we can move into Objective 3. Here we're going to discuss the chemistry of monosaccharides, including how they participate in redox reactions, how they form esters, and how they connect to other sugars through glycosidic bonds. One of the most important biochemical reactions involving carbohydrates is oxidation. Oxidation allows the body to harvest energy from sugars. In particular, aldoses like D-glucose, they can be oxidized at the aldehyde group, and they're going to produce compounds known as aldonic acids. A common example is the oxidation of D-glucose to form D-gluconic acid. Now, sugars that can undergo this type of oxidation, they're called reducing agents because they have a free aldehyde group or a group that can be converted into one. These sugars test positive with reagents like Tollens or Benedict's, which are used to detect the presence of reducing agents in solution. Actually, let's discuss the Benedict's test as an example. When beta-D-glucose is mixed with Benedict's solution, which contains copper 2 ions in basic solution, the aldehyde group at group 1 is going to be oxidized to a carboxylic acid and it'll form D-gluconic acid. During this reaction, the copper 2 plus ions, those are going to be reduced to form copper 1 oxide. This is a red precipitate that signals a positive result. So to summarize, Benedict's test, it detects reducing sugars by monitoring whether a red copper 1 oxide precipitate forms. And if it does, the sugar has been oxidized, and that confirms that it can act as a reducing agent. Now, you might be wondering, what about ketosis, like fructose? It turns out that ketosis can also act as reducing sugars, but only after they undergo tautomerization to convert into an aldose form. Once they do that, they can participate in the same kinds of redox reactions. In addition to oxidation, monosaccharides can also be reduced. If the aldehyde group on an aldose is reduced to an alcohol, then the product is called an alditol, or a sugar alcohol. Another modification involves replacing a hydroxyl group with a hydrogen and producing a deoxy sugar like deoxyribose. Now, there's another important class of reactions we want to talk about. Those are esterification. Because carbohydrates have multiple alcohol groups, they can react with carboxylic acid derivatives to form esters. Let's look at this example. Glucose here is being treated with acidic anhydride in the presence of pyridine. Each hydroxyl group here is then converted into an acetate ester to give us the following product. Now, in the body, a similar reaction happens, but instead of acidic anhydride, we use phosphates. When glucose is phosphorylated, a phosphate ester is formed. This is how glucose is trapped inside of cells at the start of glycolysis. And here's what this looks like. ATP donates a phosphate group to glucose, converting it into glucose 6-phosphate, and ATP is converted into ADP in the process. Now let's talk about one final type of reaction, and that is glycoside formation. Now this one is especially important when we start forming disaccharides and polysaccharides. Let's start from the beginning here. Hemiacetyls, like the ones found in cyclic monosaccharides, they can react with alcohols to form acetyls. Specifically, the hydroxyl group on the anomeric carbon, it's going to react with an alcohol, and that OH group is going to be replaced with an alkoxy group. And this gives rise to either alpha or beta acetyl products, depending on the stereochemistry of the substitution. The new carbon-oxygen bond that's formed, that's called a glycosidic bond, and the resulting molecule is called a glycoside. Now, for example, when beta-D-glucose reacts with ethanol, which is an alcohol, it can form either the alpha or the beta product, depending on which face of the anomeric carbon that the alcohol attacks from. These glycosidic bonds are how monosaccharides link together to form larger carbohydrates like disaccharides and polysaccharides. And with that, we can actually move into our final objective, where we are going to discuss complex carbohydrates. We're going to start with disaccharides. Disaccharides form when two monosaccharide units are joined together by a glycosidic bond, and in the process, a molecule of water is lost. Take a look at this example. When two glucose molecules link together, they form maltose. This is a disaccharide. The glycosidic bond is formed between the anomeric carbon of one glucose and the hydroxyl group of the other, with water being a byproduct. Now, there are a few disaccharides that you're expected to be familiar with, and what matters here is being able to recognize the monomers, the type of glycosidic bond, and the linkage position. So first we have sucrose. This disaccharide is composed of glucose and fructose. Specifically, it's an alpha-1,2 linkage, meaning the alpha anomeric carbon of glucose is linked to the carbon 2 of fructose. And this bond connects the anomeric carbon of both sugars, which is why sucrose is a non-reducing sugar. Neither ring can reopen. Next is lactose. This is made of galactose and glucose. Here we see a beta-1,4 glycosidic bond, where the beta anomeric carbon of galactose is connected to carbon 4 of glucose. Now, unlike sucrose, only one of the anomeric carbons is involved in this bond. So lactose is a reducing sugar. Lastly, we have maltose, which is composed of two glucose molecules that are linked through an alpha-1,4 glycosidic bond. Like lactose, maltose is a reducing sugar because one of the anomeric carbons is still free. Now, moving on to polysaccharides. These are long chains of monosaccharides that are linked together by glycosidic bonds. If the polysaccharide chain is made up of only one type of sugar, it's called a homopolysaccharide. If it's made up of a mix of different sugars, then it's called a heteropolysaccharide. In biological systems, the main sugar that's used to build polysaccharides is D-glucose. Now let's go over some important polysaccharides that you're going to need to know for the MCAT. First, we have cellulose. This is made of beta-D-glucose units, and they're connected by beta-1,4 glycosidic bonds. These linkages allow cellulose to form long linear chains that pack tightly together with strong hydrogen bonds. Because of the beta linkages, humans actually can't digest cellulose. We don't have the enzyme that's needed to break those bonds. Next is starch. This is the main storage form of glucose in plants. It's made up of alpha D glucose, and starch comes in two forms. Amylose is a linear glucose polymer that's linked by alpha-1,4 linkages. Amylopectin is branched, and the branches form alpha-1,6 linkages. Starches like amylose and amylopectin, they're broken down by enzymes in the body. These alpha linkages are digestible by humans using enzymes called alpha amylase and beta amylase. Alpha amylase, it cleaves randomly along the chain to yield shorter polysaccharide chains, maltose, and glucose. Beta amylase, it cleaves amylose at the non-reducing end of the polymer to yield maltose. Finally, we have glycogen. This is the storage form of glucose in animals. It's similar in structure to amylopectin, but with even more branching. And this high degree of branching makes glycogen more soluble in water, and it allows glucose to be mobilized more quickly when the body needs it. Enzymes like glycogen phosphorylase, it cleaves glucose units from the non-reducing ends of glycogen, and it releases them as glucose 1-phosphate so that they can enter glycolysis. And with that, that's a wrap, folks. We've completed this chapter. I hope it helps. Please let me know if you have any questions, comments, concerns down below. Other than that, good luck, happy studying, and have a beautiful, beautiful day, future doctors.